Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about another stage of translation, which we call that elongation of translation in prokaryotes. In the previous videos lectures of this series, if you guys remember, we talked about the protein synthesis overview, and we talked about the requirement which are involved in the protein synthesis, or the requirement which the cell need for the protein synthesis. And after that we talked about the initiation of translation in prokaryotes in a detail. Okay, and if you missed these video lectures, so go back to my channel and watch it. So today in this video lecture we are going to talk about elongation of translation, but before that you guys need to understand these three video lecture. Once you get idea about these three videos lecture, then elongation of translation will be very easy for you. So let me erase this one portion and let me start. So let me write elongation of translation in prokaryotes. Now in elongation of translation in prokaryotes, what we are going to talk about, we are going to talk about the introduction of elongation, and we are going to talk about the requirements which we need in elongation of translation, and we are going to see the steps which are involved in elongation of translation, and after that then we will see the mechanism of elongation of translation. Now in the previous video lecture, if you guys remember, we are at that stage that we have two subunits which are connected together, and it will make 70S initiation complex. And after that, what happened? The transfer RNA which is present at the P site carry the what we can say amino acid, which we call that F-methionine amino acid. Okay, and it will be present at the P site. Now we are at that stage. First of all, let's talk about the elongation of translation. First of all, let me talk the introduction of elongation of translation. Now, what is elongation of translation? Elongation of translation, again, it is the stage of the translation in prokaryotes, in which, not in prokaryotes, in microbes protein synthesis, in which the peptide bond are formed, in which the peptide chain are formed, sorry, not bond, chain are formed. And how it is formed? When the amino acid are connected to each other, and due to this way, when a long peptide chain are formed, and after that this peptide chain are released out, and further it is used for the protein, then it is called elongation of translation. In this case, what exactly happen? The transfer RNA which is present at the P site, then a new transfer RNA will come to bind at the A site. After that, it will also carry the amino acid which we call aminoacyl transfer RNA. So then the bond formation between the first transfer RNA which is present to the P side and the second transfer RNA which is present to the A side occur. Now after that, once the bond formation occur, so then what happen? This transfer RNA will be uncharged, and this amino acid will be transferred to the first transfer RNA amino acid. Now after that, what happen? Translocation process will occur. During translocation, what happen? The ribosomes will move and leave one codon. Okay. Now once it leave one codon, so then the uncharged transfer RNA which is present to the P site shifted to the E site, and the transfer RNA which carry two amino acid will be shifted to the P site, and again A side is free, and again the new transfer RNA come here, and what happen? Again this amino acid will be shifted to the new transfer RNA, and the process is then after that the ribosome will move again, and after that what happen? The uncharged transfer RNA will be released out, and this process is continuously going on, and this is that's why we also call that this is actually elongation cycle. Okay, so when the process is going on and repeated again and again, we call it elongation cycle also, and this process is repeated unless it reaches to the stop codon. This is the idea about the elongation of translation, introduction of the elongation of translation. Now what we required in elongation of translation, this is just simple idea. Now let's talk it in a detail. What actually, first of all, let me talk about the requirement, that what we required in this case. Let me write requirements. So in this case, we also required the 70S initiation complex, and along with 70S complex means we required two subunits of the ribosomes. We required this RNA, messenger RNA and transfer RNA. Along this, we also required the elongation factors. So let me write elongation factor. And in elongation factor, we mainly required elongation factor Tu, elongation factor G, and elongation factor Ts. Okay, so these things that we required. Okay, moreover, we also required the energy which we obtained from the GTP. This GTP will give us energy which is converted into GDP plus pyrophosphate or inorganic phosphate. So these are the things that we required in elongation of translation. Now then, how many steps are involved in elongation of translation? So let's talk about that. We also called it phase of elongation cycle. Okay, so we mainly required 3 phases of elongation of the cycle. So the first is called as aminoacyl transfer RNA binding step. The second step is transpiptidation reaction. Transpiptidation reaction. And the last step is translocation. So these are the main steps that are involved in this stage. Okay, now what happened? Let's talk the mechanism of this elongation of translation. First of all, what happened? We have this elongation factor Tu will get activated. For the first time, what actually happened? First of all, the EFTU, any elongation factor Tu is get activated. So this is elongation factor Tu. It will get activated. And elongation factor Tu along with GTP, not only this ETU, we have also GTP. Then after that, what happened? It will react with the transfer RNA. Then after that, what exactly we get? We get the structure like that. So this is the new transfer RNA. Okay, it carry the amino acid. It could be phenylaline, alanine, leucine, tyrosine, any kind of the amino acid. Okay, now it have also the anticodon, and along with this second transfer RNA having the amino acid or second aminoacyl transfer RNA, they have what? They have this elongation factor Tu, and moreover they have also this GTP. Okay, so this is the GTP. One thing that I forget, that remember this EFTU and EFG, these are actually GTPase protein. And why these are the GTPase protein? Because it will further give us protein, it will further give us energy. Now remember, when GTP is converted into GDP and pyrophosphate, then energy is released out. And this energy are then absorbed by this transfer RNA by binding to this A site. So energy is required. So this energy are then given by this EFTU. Now what happened? Once this process done, then this transfer RNA will be shifted to this area. Now the transfer RNA get activated and it will bind to that area. So this is the second aminoacyl transfer RNA which will bind to that specific area, having the anticodon. And they also carry their own amino acid. Okay, so this is the first step, which we called it aminoacyl tRNA binding. Now during this stage, what happened? Now what happened? Once it bind to that specific area, then EFTU and GTP are get released out. The main function of this EFTU and GTP will give energy to this transfer RNA, new aminoacyl transfer RNA, to just bind to that A site perfectly. Once it bind to this A site perfectly, then these two proteins are get released out. And what happened? Once these two proteins are get released out, let's suppose the EFTU and GTP are get released out. Then this GTP will convert it into GDP. But how it will convert into GDP? What happened? We have another elongation factor which we call a elongation factor TS. So elongation factor TF will convert the GTP into GDP plus pi, means inorganic phosphate or pyrophosphate. Once we get this thing, then energy will be released, and this energy are then used by another transfer RNA. Then once the next transfer RNA, which is the third aminoacyl transfer RNA, is used this energy, then again EFTU will bind and GTP will bind, and it will set this new aminoacyl transfer to this site. Okay, so the cycle is repeated again and again. This is the first cycle. Okay, next what happened, the transpiptidation reaction will occur, and it will mainly done by this 50s of 23 sRNA. So inside the 50s, what we have 23 sRNA. So there we have 23 rRNA, and it will actually perform the transpeptidase reaction activity. That's why we call it transpeptidase enzyme, and that's why we also called it ribozyme. During this stage, what happened? The bond formation will be occur between the first aminoacyl transfer RNA and the second aminoacyl transfer RNA. Means the transfer RNA which is present at the P site and the transfer RNA which is present at the A site. The bond formation will be occur in each amino acid. Now how? For example, this is the first amino acid. So it have the amino group. Okay, and this is the second amino acid, let's suppose, and it have the carboxylic group. So this amino group, which we called it alpha amino group, it will attack to this carbon, which we called alpha carboxylic carbonyl group. Okay, so once the attack is done, so then what happened? The bond formation will occur. Okay, so then the bond which is present between this transfer RNA and the amino acid are break down, and the bond will be formed between them, as you can see. So it means that the amino acid which is present with the transfer RNA of the P site will be shifted to the transfer RNA of the A site, and thus we get two amino acid, and this is actually peptide chain. Not a peptide chain, but somehow we can say a peptide chain are formed. Now remember guys, in this case no energy is required. Why? Because the energy is present between this transfer RNA and the amino acid. There is also a bond present. So in this case, when the amino acid are attached to each other, as you can see, so during this stage energy is not required. Why energy is not required? Because the high energy is already stored in this transfer RNA. In this transfer RNA and in this amino acid, energy is already stored. That's why energy is not required. So this is the second elongation cycle of this transpeptidase reaction. Now after that, the next stage will occur, which we called it translocation. Okay friends, now what happened in translocation? In translocation, the ribosomes will move in the 5' to 3' direction. Now when the ribosomes move in a 5' to 3' direction, it means their protein are synthesized in 5 to 3' direction. Okay. Now when the ribosome move in 5 to 3' direction, then what happen? It leave one of the codon. Now remember, if it move, so definitely one codon will be left, and the transfer RNA which is present at the P site, which we call it uncharged transfer RNA, because the amino acid are cleaved, it will be shifted to the E site. And the transfer RNA which is present at the A site, which carry the two amino acid or which carry the peptide chain, will be shifted to the P site. Then after that, A site will get free. So what happen if I draw the same structure here again? So the situation will be like that. So this is the previous one codon. So it is left by this ribosomes. Okay. So now remember, this is the small subunit. It is now shifted. So when it's shifted, so it will leave one codon, as you can see. And this is the large subunit of the ribosome. And again, they have these three sites, as you can see. Okay. And what happened? This transfer RNA will be shifted to this position. And this transfer RNA having the peptide chain will be shifted to this position. Okay, so this is called translocation. And again, the A site is free. Now, how this process is done? This translocation will be done by an enzyme which we call that translocase. And this translocase enzyme is just like EFG, elongation factor G. So elongation factor G is acting as a translocase enzyme, and that's why this process is done by this enzyme. Now, if we talked about this enzyme, so remember in this case what we have, this is, let's suppose, EFG. So remember EFG, in EFG what we have GTP. Okay, so this is EFG. So this GTP is again used in GDP plus pi. Pyrophosphate is released out, and this energy is now used by this ribosomes for their movement. And once the ribosome moves, so then the tRNA peptide bond which is present to the A side will be shifted to P side, and the uncharged tRNA will be shifted to E side. Again, the A side is freed, as you can see. Then after that, new transfer RNA will get activated, and it will proceed to the same step as we talked for this transfer RNA. So again, this is the new transfer RNA. Now it will comes and bind here. Means elongation ETU plus GTP, and when these two things are combined together with aminoacyl transfer RNA, then it will brought this transfer RNA to the A site. Okay, now once it brought this transfer RNA to the A site, so then these two factors are released out. This GTP is then, what happened, we talked that this GTP is then converted into GDP and plus inorganic phosphate by the help of elongation factor TS. Okay, now this is used for the energy. Now this is actually energy is released, and this energy is used for the next transfer RNA, for the next transfer RNA activation. After that, what happened, we talked then this is the first step. Then in the second step we talked that transpiptidation reaction will occur, means the bond formation will be occur, and the bond formation will be occur like that manner as we talked. So there will be a bond formed, as you can see. So now the transfer the amino acid which is present with this transfer RNA are cleaved, and it will be shifted to this new transfer RNA, to this third transfer RNA, we can say like that. Then after that, what happened, the translocation process will occur. So this ribosome will move. Now remember, when this ribosomes will move, translocation process when occurs, so then after that what happened, again we can say that the tRNA which is present at the E side will be released out, and the tRNA which is present to the A side will be shifted to the E side, and the tRNA which is present to the A side will be shifted to the P side. So after that, what happened, this is the first codon which is left by the ribosome. Now this is the second codon which is left by this ribosome, and again this is another codon. So now the ribosome is at that place, as you can see, and this is the large subunit. So what we have again, so this is the tRNA which is present as the E side will be released out. So this is the tRNA, uncharged tRNA are now released out, and this tRNA which is uncharged will be shifted to the E side, as you can see. During translocation, it will be shifted to the E side, and this transfer RNA which carry the 3 amino acid, or we can say which carry the peptide chain, will be shifted to this P site. Okay, and thus A site is again freed, and then new transfer RNA will come, and the same process will be repeated again and again. So we can say these 3 steps is repeated again and again until and unless when this ribosome move to the stop codon. As you can see, it leave one codon, and this process is going on, and it leaving one codon at a time, and due to this way we get the long chain of amino acid, as you can see. Now when we get a long chain of amino acid, so then this ribosomes will move at a specific codon which we called it stop codon. So at stop codon, the ribosome will be stopped and no more peptide chain will no more synthesized. And at that point, translation will be stopped and the ribosome will be separated from each other. But we will talk it in termination of translation in detail. Okay, so this is in a sense about elongation of translation in prokaryotes. I hope you get idea about that. Inshallah, in the next video lecture, we are going to talk about termination of translation in detail. So please stay tuned and keep watching. Thank you so much for watching this video.